Now I'd like to introduce the chair of Vision Quest 2012. Our first speaker this morning, Dr. Ian MacDonald. He's the professor and chair of ophthalmology at the University of Alberta. He's an ophthalmologist and a geneticist. He's internationally known for his research on choroideremia, a form of RP, including the development of a genetic test that's now used to diagnose choroideremia. I'm proud to say, and this is a very proud moment for our organization, that Dr. MacDonald and his colleagues at the University of Alberta were recently awarded a $1.3 million grant from, which was partnered and jointly funded by the Canadian Institutes of Health Research, the Choroideremia Research Foundation of Canada, and the Foundation Fighting Blindness Canada. Congratulations, Dr. MacDonald.
Thank you very much, Sharon, for those kind words, and thank you as well as a community for your support for our trial. I'll talk a little bit about that. And thank you, Sharon, for all your hard work on behalf of Canadians with vision loss and championing vision loss as an important part of the healthcare of all Canadians. Really, it's a true pleasure to be here, to be the chair of Vision Quest, who have been asked. It's not about me, it's about all of this community. We have a wonderful community of researchers here in Edmonton that are working on vision loss, and also in Calgary, and so it's nice that we can share all of our thoughts with you and our hopes for the future. I'd also really like to thank all of these people: Andrew Washevitz, Arden Lehman, Carol Shermans, Tanya Bubala, Mark Grieve, Yves Sauvé, and Matt Tennant for taking time to come and speak about their knowledge and impart their knowledge to you. And also like to thank the sponsors: Novartis, Bayer, Medicis, Sick Kids, AMI, and the Alberta Society for the Visually Impaired, and bringing your materials too that you can share with us. And the CNIB, who have been champions along the way, all this way. Well, and gosh, our volunteers, it's nice to see these happy faces who are keen. Well, the first thing I saw when I was walking down was seeing a nice young volunteer helping somebody in, and it's so nice to see that. Wonderful experience for everybody. I was thinking back that in my own childhood and as I was growing up, I don't think I had really had a true appreciation of what blindness was. And but now I really do understand, I think, as much as I can, what blindness means. So there have been some amazing advances in my career in terms of advances on our understanding of blindness, the genetic causes of blindness, and the possibilities of treatment. I started working on this disorder called choroideremia about 30 years ago, and there was a lady who was at the Ottawa Citizen who phoned me and said, is anybody doing any research on choroideremia? And I said, I'm not aware of any, but would you like to get started? And that was 30 years ago, so you can imagine the time frame of vision research. And Sharon was right in pointing out that we have made some significant advances in the study of choroideremia, first isolating the gene, understanding the genetic cause in that gene of causing choroideremia, developing a test for choroideremia, a test that's relatively simple, taking the protein from cells in the body, seeing that protein is missing in individuals with choroideremia, and now the opportunity really to try a gene therapy experiment. So, genetics definitely is my area of expertise, but I'm humbled by the complexity of genetic disease. There are now over 200 genes that have been identified as causes of heritable blindness. But knowing these genes and which ones can cause blindness can help us diagnose conditions more quickly, and also accurately allow us to counsel patients and families. And technology has really advanced in a terrific pace. When I first was doing gene research 30 years ago, I was amazed that you could isolate DNA from cells, put it in some chemicals, put it in a test tube, put a glass rod in it, and then bring this string of material out of the glass tube called DNA, and then use enzymes to break up the DNA and then run it on a gel and isolate different segments of the DNA. But in those days, we had to use radioisotopes, nasty things, and it would work behind plexiglass containers, and you would be told, oh yes, it should be okay, it doesn't penetrate more than skin deep. So it should be okay. And you sort of wondered about your hands. I didn't think of myself as Marie Curie, but you sort of wondered about that. Well, we've gone beyond that now. We don't use radioisotopes anymore. We use different things, fluorescent probes that actually allow us to test the DNA, sequence the DNA, find out where those little changes are in the DNA. Wonderful technologies. Things that are called arrays, where we can take a glass slide and put these tiny little molecules of DNA on the glass slide and then match your DNA with the DNA that's on this glass slide and say, aha, there's where the genetic change is. Marvelous technologies that we wouldn't have ever conceived about 30 years ago. So knowing which genes are involved allows us to understand what goes wrong in the retina and helps us as well to develop treatments. And I wanted to give you the example of the QLT trial in Montreal. It's being led by Rob Kunakoop, who's a researcher that's had Foundation Fighting Blindness support for many years. And he is involved in a trial of a therapy for Leber's congenital amaurosis. It's an infantile form of retinitis pigmentosa. And I'll take you now to Calgary, where I saw a young family with a young boy who was about 3 or 4 who had a condition that looked like Leber's congenital amaurosis. He couldn't see very well, he couldn't see in the dark. What was very interesting, though, when I looked into his eyes as an ophthalmologist, I could see that his retina appeared absolutely normal, but his electroretinogram was severely impaired. And it classified him, it gave him the label of Leber's congenital amaurosis. And I thought, well, okay, so Leber's congenital amaurosis isn't just one disorder. It's probably at least 12 or maybe 13 different genetic diagnoses. So we must find out what the particular cause of this disorder in this boy is, because it might possibly lead to a treatment. Even I was a little bit skeptical at that time. Well, you know, if we find the gene, where do we go from there? So we undertook a very complex genetic analysis looking at the inheritance pattern of this particular disorder in this boy, and then found a gene called LRAT. What an awful name, hey? LRAT. And it's a gene that's involved in the biochemistry of vision. And both the LRAT gene and the RPE65 gene, when changed in 2 copies, can result in a form of Leber's congenital amaurosis that is amenable to biochemical therapy. And Rob Kunakoop with his team in Montreal have a clinical trial on the treatment of those 2 conditions. And so the family packed up, took a plane, went off to Montreal, and they had this treatment. It's an oral treatment with a vitamin A-like compound, and miraculously, and I really mean that, his visual function improved and has maintained that stability of improvement over the past year, and will probably continue to follow him. And I thought to myself, gosh, I don't think I've ever seen that before. I don't think I've ever seen where you take this genetic technology, find the gene, and then take it to treatment and then improve somebody's vision. It was an outstanding example for me personally that the promise, the hope that genetic technologies will lead to treatments is actually valid, that we will actually find these genes, find the treatments, and then go from there. So there are other therapies that are coming forward. You've heard through Sharon about this potential upcoming gene therapy trial in Canada on choroideremia. For me, it's very important to bring this to the community as we've worked on this for many, many years. But what's also exciting is that we're developing the actual format, the venue, the expertise, the people, the machinery to actually undertake gene therapy trials on other conditions. So it's not just about choroideremia, it's about bringing other therapies on other heritable forms of blindness as well. So let me describe briefly how gene therapy works. We have to have a very sophisticated lab that makes a vector. We call it a vector. It's the virus that's been packaged that's non-pathogenic, and that's going to be the carrier of the gene into the cells of the body. But how do we get that to the eye? Well, there are two vitreoretinal surgeons who are going to be joining us today, Matt Tennant and Mark Grieve. They have, over their careers, developed very sophisticated technologies where they can actually manipulate the retina, admittedly under an anesthetic, so you don't have to worry that you're awake when this is happening. But we would predict that the surgical procedure would occur under probably an anesthetic, where we remove the vitreous, which is that clear jelly at the back of the eye, expose the retina, and make a small puncture in the peripheral part of the retina, and then blister the retina, make a little bubble of fluid, and then inject in that fluid the viral vector that has the gene. And through the normal physiology of the eye, the retinal pigment epithelial cells that are underneath begin to pump out that fluid, and then the virus gets absorbed into the photoreceptors and the retinal pigment epithelium, and then they take on that gene as theirs and say, aha, okay, that's part of what I've got. And they start producing the normal gene product. In this case, it might be choroideremia, if we were doing that particular gene. What's fascinating is that it's not just choroideremia that the world is interested in. You're quite aware of the Leber's congenital amaurosis trials in Europe and the United States that have shown that this approach is not only safe, but also effective, that individuals have repeatedly had their vision improve with gene therapy for the RPE65 form of Leber's congenital amaurosis. And that's work that has been done for many years in the UK, Philadelphia, and Naples, Italy. But now we have trials that have started. Patients have undergone therapy for Usher's syndrome due to mutations in the myosin 7A gene. And also there's a trial that is planned on Stargardt disease. And I know that I'm not knowing exactly what's going on everywhere, but it's wonderful to see this bloom of activity across the world. We're almost there with our gene therapy trial here in Edmonton. We still are awaiting our Health Canada approvals, and so I can't say when we're going to start, but I would very much hope that by the beginning of next year, in the spring, that we're able to undertake these studies. So gene therapies are very specific. They're trying to stop the death of retinal cells, and there are many ways of approaching this, and you're going to hear a little bit from Dr. Lehman about, I hope, his studies on a gene defect that causes LCA and the possibility of a drug treatment for that, a new drug, P7C3. There are therapies beyond gene therapy as well that are on the horizon, and these involve devices. Some of these devices require retinal surgery and the implantation of a retinal array on the back of the eye that will, in order to capture light. These are retinal implants. And there are also individuals who are studying introducing genes that will allow other cells in the retina to start functioning as photoreceptors. There's some remarkable work by Gautam Awatarami at the University of Victoria, who's looking at probing circuits in the retina, trying to regain the retinal function and the retinal circuitry that is important. And this is work that's being done in mice at the present time, but you can imagine that many of these studies will begin to be translated to human studies over the next years. And Carol Sherman is going to talk to you about stem cell therapy, and I would be very much looking forward to those talks and understanding a little bit more about how stem cells can act in a reparative fashion to restore vision and improve visual function. So I haven't really touched on everything. It's a very broad international community that's working on these. I was asked last night, well, are you well connected, Dr. MacDonald? And I think this is a very, very, very important question. So who I know is very important, because if I don't know something about something, I can reach out to that community, ask people in London, England, what do you know about this? Do you think this might work? And I would reassure you that that dialogue is active, and sometimes it's hard to keep up with the email traffic, but we do talk internationally. There is a bit of trying to get there first, but I think we've learned a lot that it's not getting there first and taking credit for that. It's really trying to help people restore their vision, and it's not all about getting there first. So I hope you really enjoy the day. Ask lots of questions. You've got some experts in many, many, many different fields to ask about the different things that you're concerned about. Scientists really enjoy these events because they can show off their knowledge base, and you can test them. And they'll come away with thoughts and ideas that maybe they hadn't thought of it that way. So you can send some stumpers out there too. So thank you for coming, and I hope you find it an inspiring and informative day, and I do believe that I've left enough time for some questions. So John, maybe you can ask the first question.
All right. So my question is around a term called bottlenecks. And as far as the research community is concerned, where are the bottlenecks right now? Is it a technology bottleneck? That is, we don't have the technology to implement or to test or to progress sort of the biochemical, the physiological type of, or where's our bottlenecks in the implementation? Like, where are we stuck in our progress?
Right. So that's a very good question. So let's take it all the way to Health Canada, where you have new therapies that are arising, either drug therapies or new devices. It may seem like a bottleneck, but it's there as a protection. We can't have therapies that are not proven in some fashion arising. And maybe they're bottlenecks, but I think they are safety mechanisms that need to be there to make sure that we are approaching these things carefully, that patients aren't rushed into therapies with false hope. I would hate to say it's a bottleneck, but it is a regulatory requirement. For example, when we undertake a clinical trial, we have Tanya Bubala, who's, I think, going to talk quite a bit about this in one of the breakout sessions. We need to go through a process of asking for approvals. And I think these, to me, as a researcher, do create some bottlenecks because we have to educate and convince people beyond us. The researcher and the, let's say, the research subject are very keen to move ahead. And I would perceive that they both think of the regulatory bottleneck as an impediment. It just seems to be taking a long time. And one of the other bottlenecks, I think, is funding. We'd very much like to undertake some of these studies, and I think it's a great privilege for our group to have been selected to do some of this work, but imagine that there are other very important formative ideas that aren't being funded simply because there isn't enough money to go around. We live in a very changing environment where demands for funding are very great, and so maybe that could be a bottleneck as well.
Hello, Dr. E. MacDonald.
Nice.
I'm a client of yours. My name is Judith Tomiak.
Nice to see you.
And I'm really excited about this conference today. I'm just a little bit nervous. I just wanted to let you know that you are my best friend, and Foundation Fighting Blindness and all you researchers are my best friends right now because I'm starting to lose my eyesight. More. I'm 62 now, and I could see really well for the last, since I was about, when I was diagnosed when I was 19. I drove until I was 40. And so now with my last round of eye tests, I'm told that there is nothing more they can do for me. I have 5% vision left, and I'm really going through a hard time right now. So you are my best friend. I wanted to know, my question today was, through all my sentiments today, is when you're ready to do clinical trials, I want to be the first one there. And how do you let people know when you're ready to do that?
Right. So thank you very much. She has lovely warm hands, too. One of the key elements for letting people know about clinical trials is something called the web. And it's an amazing resource. I got on the web this morning just to familiarize myself with what's there. So the Foundation Fighting Blindness, as Sharon said, creates an inventory of what research is ongoing. And gosh, you could listen to these wonderful audios of the previous meeting in Newfoundland, and I would point to those. So that's one resource that you can go to, and you can see what's happening, when's it coming to my front door. The other thing that you can look at is a website called clinicaltrials.gov. It's www.clinicaltrials, all one word, .gov. And you can type in your diagnosis. And then you can find out what's going on, not just in Edmonton, but everywhere in the world, any reputable clinical trial that has ethical approval. And then you have their contacts, how to get in touch with them, what's the telephone number. And this is brought to you by that wonderful government that's just south of us called the U.S. government, Health and Human Services. And they do some wonderful work. Does that answer your question?
It does. I just wanted to let you know that I also have a brother who's totally blind and a younger sister, and they don't really care like I do. I really care. I care for me, and I care for them, and I care for my whole deafblind community, that they not give up.
We're with you on that one.
Thank
Very
you,
much
Dr.
so.
MacDonald. Thank you.
Thank you.